So my name is Elizabeth Topp, and I am the director of the William D. and Sherry L. Young Institute for the Advanced Manufacturing of Pharmaceuticals at Purdue University. So the Pfizer and Moderna vaccines for the COVID pandemic are mRNA-based, so that's messenger RNA, and it's the counterpart to DNA in our cells, right? So you know that DNA makes RNA, makes protein, is the fundamental hypothesis of biology. So we've started making mRNA vaccines, and we all learned that when the COVID pandemic hit and the Pfizer vaccine came out first, we all saw those freezer farms, right? Yards and yards, rows and rows of -80 freezers keeping the vaccines ultra cold. And I'm sure people in the general public, and I did too, say, what the heck is going on here? Why are we generating these freezer farms for this mRNA? The difference between the structure of DNA and the structure of RNA has to do with a single oxygen atom on a ribose sugar subunit of the RNA that's repeated over and over again in the molecule. And that's the thing that's different, among other things, between RNA and DNA. Because of that oxygen, RNA has to be stored at -80 or the vaccines would never be intact by the time they got around the world to people who needed them. One of the ways that we try to deal with the problem of having drugs fall apart is by making them into solids. And so sometimes we can do that just by drying them, by making them into tablets and capsules, for example. Often those forms are more stable than solutions. And a lot of the drugs you see at Walgreens or other kinds of drugstore chains, a lot of the drugs that you have in your home are probably in those forms. But we also make solid forms of drugs that are meant for injection. And in many cases, whether they're over-the-counter or prescription medications, we often make them in solid forms because the solid state is more stable than the solution state. If we talk about technology to make a more stable mRNA, then obviously one of the things that we're thinking about are mRNA vaccines. Some of the degradation reactions that we care about a lot for drugs are what we call hydrolysis reactions, and those are reactions with water. If you make solids, you take the water away and therefore slow those reactions that happen with water. The subtle difference in chemistry makes a big difference in how that molecule degrades. Understanding how the molecule degrades and what the pathways are means that we might be able to do something to stop it from doing that. And so the frontline mRNA vaccines, as we all know, were used for treating COVID. And so they're the ones that kind of kick down the door to mRNA vaccines of all kinds. Other kinds of mRNA vaccines include vaccines for cancer. And so it's weird sometimes to think about cancer vaccines, but we do have cancer vaccines that treat cancer, not that necessarily prevent cancer, but that are treatments for cancer. And then that molecule class, that mRNA class, is related to a whole broader set of molecules that have that same RNA type structure. Some of them are quite small, call them oligos or oligonucleotides. And then there are others in that class that are sort of medium-sized. And so if we figure out how to stabilize the big ones, then those same technologies ought to be useful for some of the small and medium-sized ones too. And they treat all different kinds of disease states, but two big ones are infectious disease and cancer.